Hey everyone, welcome to the channel. If you want to ace your Class 9 Science, Social Science, English and more, you are in the exact right place. In this video, we are diving deep into Chapter 2 of Science, Cell, the building block of life. I am going to make sure you understand every single topic, from thermophiles all the way to programmed cell death, using super simple words and real-world examples. These videos are going to be your ultimate study companion, so don't forget to subscribe, like, share, and watch until the very end. Thermophiles. Simple explanation. Imagine taking a bath in water that's literally boiling hot. You'd get burned, right? But there be tiny single-celled organisms that actually love this extreme heat. They be called thermophiles. Thermo means heat, and phile means lover. They live in scorching hot springs, like the Puga Valley in Ladakh, where the water's near boiling point. Real-world example. Think of thermophiles like the ultimate stunt performers of the microscopic world. While most living things need a cozy, mild temperature, these lads are perfectly happy living inside nature's pressure cooker. 2. Limit of resolution and Robert Hooke. Simple explanation. Our eyes are great, but they have a limit to how close two objects can be before they blur into one single dot. This boundary is called the limit of resolution. For humans, it's about 0.1 millimeters. To see anything smaller, we need help. Back in 1665, a scientist named Robert Hooke built his own early microscope. He looked at a thin slice of cork from tree bark and saw tiny empty boxes that looked like the small rooms monks lived in. He named these boxes cells. Real-world example. Imagine looking at a digital billboard from a mile away. It just looks like one big image. But as you walk closer, you start to see the individual light bulbs, pixels. Robert Hooke was the first person to step close enough to see the pixels of life. 3. Magnification. And all about microscopes. Simple explanation. To see cells, we use magnification, which means using lenses to bend light or electrons to make tiny objects look massive. School labs use light microscopes, which use glass lenses and light to zoom in up to 40x or 100x. But when scientists want to see the superfine details inside a cell at a nanometer scale, they use a massive, powerful machine called an electron microscope, which uses a beam of electrons instead of light. Real-world example. A light microscope is like using a standard magnifying glass to read tiny text. An electron microscope is like using a high-tech satellite camera that can zoom in from space to see a coin on the ground. 4. Structure of a cell and the cell membrane, plasma membrane. Simple explanation. Every cell has a boundary that holds everything together, called the cell membrane or plasma membrane. It is selectively permeable, meaning it acts like a very strict security guard. It lets good things, like food, oxygen, and water enter, but keeps bad things out and stops the cell's insides from spilling away. Fluid mosaic model. The structure of this membrane is explained by the fluid mosaic model. It's made of a double layer of fats, lipids, with proteins floating around in it. It's called fluid because the molecules can slide, flip, and move sideways, and mosaic because the proteins look like decorative tiles scattered across the surface. Real-world example. The cell membrane is like the walls of a house, and the proteins are the doors and windows. The walls protect you, but the doors and windows selectively choose who or what gets to walk inside. 5. Osmosis and diffusion. Simple explanation. How do things move across that cell membrane? Diffusion is when particles naturally spread out from a crowded area to a less crowded area, until they are even. Osmosis is a special type of diffusion. It is strictly the movement of water across a membrane. Water always travels from a dilute side, lots of water, little salt, sugar, to a concentrated side, less water, lots of salt, sugar, to balance things out. The types of solutions. Isotonic. The water concentration is identical inside and outside the cell. The cell stays perfectly normal. Hypotonic. There is way more water outside the cell than inside. Water rushes in, causing the cell to swell up like a water balloon. Hypertonic. There is more salt sugar outside, meaning less water. Water rushes out of the cell, causing it to shrivel up. Real-world example. Remember the potato experiment from the text. When you put a potato slice in plain water, hypotonic, it drinks up the water. 6. The cell wall. Simple explanation. Plant cells, fungi, and bacteria have a bonus layer outside their cell membrane called a cell wall. It is made of a tough carbohydrate called cellulose. Because plants can't walk away to hide under an umbrella when it rains or storms, they need extra armor. The cell wall is completely rigid, giving plants their shape and allowing trees to stand tall against the wind. Real-world example. If the cell membrane is like a soft balloon holding water, the cell wall is like placing that balloon inside a sturdy cardboard box. Even if the balloon loses water and shrinks inside, the box keeps its strong shape. Seven. Prokaryotic and eukaryotic cells. Simple explanation. Cells are divided into two main categories based on how organized they are. Prokaryotic cells, primitive. These are simple, tiny cells, like bacteria. They don't have a protective wall around their DNA. It just floats freely in a messy clump called a nucleoid. They also lack internal rooms, organelles. Eukaryotic cells, advanced. These are larger, highly organized cells found in plants, animals, and humans. They have a beautifully protected nucleus for their DNA and lots of specialized internal compartments. Real-world example. A prokaryotic cell is like a one-room studio apartment where the bed, kitchen, and desk are all in one open space. 8. Nucleus. Simple explanation. The nucleus is the absolute brain of the eukaryotic cell. It holds the DNA, which contains all the hereditary codes and instructions for making you, you. Inside it sits the nucleolus, a busy factory area that helps build parts for ribosomes. The DNA is normally a loose, messy pile of threads called chromatin, but when the cell prepares to divide, it neatly winds up into tightly packed packages called chromosomes. Real-world example. The nucleus is the master control room, or the main office of a factory. The DNA is the giant blueprint instruction manual locked inside the office safe, ensuring the factory runs perfectly.
The manufacturing and shipping system. 9. Ribosomes, the protein factories. What they are: tiny structures floating freely in the cell's fluid cytoplasm or stuck to the endoplasmic reticulum. They are the factory workers that build proteins. Real-life example. Think of them like 3D printers in a factory. They take raw digital instructions, RNA, and assemble physical parts, proteins. 10. Endoplasmic reticulum, ER, the manufacturing plant. What it is: a massive network of tubes and sheets connected to the nucleus. Rough ER, RER. Bumpy because it is covered in ribosomes. It focuses on making and exporting proteins. Smooth ER, SER. Looks smooth because it has no ribosomes. It makes and stores fats, lipids, and hormones. In your body, your pancreas produces insulin, a protein, to regulate blood sugar, so pancreatic cells are packed full of rough ER. Meanwhile, liver cells contain a lot of smooth ER to help process and break down fats and toxins. 11. Golgi apparatus, the packaging and shipping center. What it is: stacks of flattened, sack-like pouches that act like the cell's post office. It modifies, sorts, and packs proteins and fats into tiny bubbles called vesicles to ship them out. Real-life example. The Golgi apparatus is exactly like an Amazon fulfillment center. It takes the raw products manufactured by the ER, wraps them in protective packaging, vesicles. 12. Lysosomes, the cleanup crew. What they are: Tiny, membrane-wrapped bubbles filled with powerful digestive enzymes that break down waste, old cell parts, and food. Real-life example. They function like mobile recycling trucks equipped with paper shredders. They drive around the cell, pick up broken machinery or cellular garbage, shred it down into raw materials, and dump it back out so the cell can reuse it. In your body, when white blood cells engulf a harmful bacterium, they send it straight to the lysosomes to be dissolved and destroyed. 13. Mitochondria. The powerhouse. What they are: double membrane structures that break down nutrients to generate energy, storing it in a tiny battery molecule called ATP. Real-life example. Mitochondria are the power plants of the cell. Just like a coal or nuclear power plant converts raw fuel into electricity that powers your house, mitochondria convert glucose from your food into ATP electricity to power your body, in your body. 14. Plastids, the solar panels and storage units. Plants have unique structures called plastids that come in 3 types. Chloroplasts, the kitchen, green structures filled with chlorophyll that catch sunlight to make sugar, food through photosynthesis. Real-life example. These are solar panels connected to a sugar cotton candy machine. They trap sun rays and use that energy to create food out of thin air. Chromoplasts, the color advertisers, filled with yellow, orange, or red pigments to give bright colors to petals and fruits. Leucoplasts, the pantry, colorless storage units that hold on to reserves like starch and oils. 15. Vacuoles, the storage tanks. What they are: storage spaces in a cell. Plants have one huge central vacuole while animals have tiny temporary ones. Their job: they store water, nutrients, and waste. Real-life example. Think of a plant vacuole like a water balloon inside a cardboard box. How it works. When the balloon is full of water, it pushes against the box and keeps it stiff and upright. If the plant runs out of water, the balloon shrinks and the plant wilts. Meiosis, the diversity maker. What it does occurs only to create reproductive cells, sperm and egg, cutting the DNA/chromosome count exactly in half across 4 new cells. Real-life example. Think of this like shuffling 2 decks of cards together and dealing out half hands. Because the cards are shuffled and split, every single hand dealt is completely unique. Why it matters? Because sperm and egg cells only carry half the DNA. When they combine, the child gets a fresh, unique 50/50ths mix. This is why you might have your mother's eyes and your father's hair, but you don't look exactly like a perfect clone of either one. Cell theory is the basic rulebook explaining how all living things are built. It was put together by a few key scientists who discovered different parts over time. Robert Hooke, 1665, discovered the very first cells while looking at a slice of cork under a microscope. Matthias Schleiden and Theodor Schwann, 1838-1839, discovered that all plants and animals are made of cells. Rudolf Virchow, 1855, discovered that all cells come from older cells by dividing. The 3 core rules of life. Rule 1. All living things are made of cells. From tiny ants to giant whales, everything alive is built out of cells. Rule 2. The cell is the basic unit of life. It is the smallest individual piece of an organism that can perform all life processes like eating, breathing, and growing. Rule 3. All cells come from pre-existing cells. Cells never appear out of nowhere. They are only created when an older cell divides. Thank you for watching. If you want more such amazing content about science from SS English with the help of these videos, don't forget to like, share, and comment. See you.